So I'm going to talk about hybridization in California oaks, which are sort of, oaks in general have been a sort of a classic example of hybridization and some difficulties with species concepts and with sort of understanding lineages and movements of genes and lineages. I'm going to start by sort of going back to this guy, Lee Van Valen, sort of a classic, one of like the great evolutionary and ecological theorists of the 20th century. He's very well known for sort of originating the Red Queen hypothesis, maybe less well known for introducing the ecological species concept. He never called it a species concept, he just talked about ecological species. I think the way we think about ecological species concept today is probably misrepresenting Lee Van Valen's ideas about it. If we talk about the ecological species concept at all, it's really just sort of on this list, on the checklist of species concepts, and nobody really describes what it means to be an ecological species. If anybody does think about it, they probably just sort of post hoc come up with ideas about how ecologists would think about species. And in that case, they probably think the ecological species concept is equivalent to maybe like a functional group or a guild or something like that. It's definitely not the way Lee Van Valen thought about it. Lee Van Valen probably thought of ecological species as sort of a borderline cases where other species concepts didn't work so well. We could look at the ecological context and try to understand how lineages were interacting with each other. And in that sense, the ecological species concept in the paper that Lee Van Valen wrote and introduced it was really just an introduction to this idea of a multi-species complex. A multi-species complex is a group of interacting ecological species that in some ways are distinct lineages, but because they interact and hybridize with each other, they can also be thought of as forming a single complex lineage. So Lee Van Valen's idea about this is that this ability for a multi-species complex to share alleles, to share genes, is itself an evolutionary adaptation. This is an advantage for species within this complex to be able to get alleles from other species, since they're all ecologically distinct from each other in some sense. Especially, he says here that sort of wherever they originate, they can be moving to other species. So he's thinking of it sort of in terms of new alleles arising. I think we can also think of it in terms of unstable environments. So if climate is changing, it can be very helpful to be able to get genes from another species that may be already better adapted to the climate that's sort of coming in your region. So this is very cool. This is a really cool idea. How can we separate this idea of a multi-species complex from other types of hybridization that we know happen? For instance, most common sort of ways people think about hybridization are sort of recent recontact involving sort of anciently divergent lineages, or this sort of famous gray zone. It's sort of this sort of temporary position that divergent lineages go through as they're speciated. But that's not what we'd be seeing here. This idea of a multi-species complex is not similar to either of those. So these are really three different ways of thinking about hybridization. So let's sort of imagine what a multi-species complex would look like on a phylogenetic tree. So first of all, the three lineages, or the lineages, should be ecologically distinct from one another. They shouldn't sort of have very strong overlap in their ecological traits. So I'm showing that here with three different colors to show that we're really dealing with three distinct lineages, ecologically distinct lineages. And then second, third, we should find introgression sort of throughout the phylogeny. It shouldn't just be sort of towards the tips, which would more probably indicate recontact. And it shouldn't just be sort of deep down in the phylogeny either, which would probably be the gray zone. It's difficult to say whether this is sort of the entire thing is the gray zone. You get into this problem with scale, that maybe this whole phylogeny that I've drawn here is a gray zone, and how do we know when we've left the gray zone? I don't know. Hopefully we can sort of date that phylogeny and say that if something is 5 million years diverged, that's a really long gray zone. And is there really a distinction between the multi-species complex and the gray zone? I kind of think of a multi-species as lineages that entered a gray zone, realized it was really advantageous for them, and just decided not to leave. So I'm going to be sort of looking at this and testing this idea with California oaks and trying to see if this multi-species complex applies in California oaks. So I'm going to be looking at this Western North American clade. This is a monophyletic clade, although I'm only looking at some species within it. First, Quercus garryana, the Oregon white oak, has this distribution from up in British Columbia down into Northern California, the only native oak here in Portland. Second species is California blue oak, which has this famous bathtub ring distribution around the Central Valley in California, so it's found in the foothills and the coast ranges, but not right in the valley. And then third, although I'm not going to be talking about it very much because I don't have sequence data from it yet, a third species is this Tucker's oak, which is a shrub oak, an evergreen shrub oak. That's sort of down in Southern California and into Baja. So first of all, the first thing we can say is that, and I don't have quantitative data to show this, but hopefully you'll just believe me, that these are ecologically distinct species. Life history traits are different in this one. The two trees are sort of both deciduous trees, but blue oaks are much more drought adapted. They're found in some of the driest sites in California, whereas Oregon white oak is found only in the wettest sites in California and then up into some very wet parts of the western US. Interestingly though, wherever they overlap, they're known to hybridize. So I've shown here overlapping between Garryon and the glassii in orange is a named hybrid, Quercus eplingi. And then overlap between Tucker's oak and blue oak, sort of in the southern part of the blue oak range, is another named hybrid, Quercus x albergiana. So my sampling is sort of within this area, although it's a little bit more restricted than I wish it was. I haven't gone very far into Tucker's oak, and I haven't gone very far up into Garryana. I sort of focus on blue oak, and sort of just getting just outside the range of blue oak into nearby populations of those other species. So just quickly, because it's boring, I'm going to go through sort of what the lab part of my work is. So I did RAD-seq, double digest RAD-seq. I was able to align to a reference genome. I feel lucky that since the time I started my grad program, 2 reference genomes have come out in white oaks. So I'm able to align my reads to a reference genome. And then I do a bunch of filtering. So I have like 170,000 SNPs after aligning to a genome. But after sort of filtering for some things that you can't read on there, so I'm filtering for SNPs that are present in at least 75% of my samples. I'm also filtering for only 1 SNP per contig in the reference genome to get as close as I can to unlinked SNPs. And that gets me down to like 2,000, which doesn't sound like a lot in like sort of the genomic age, but it's more than I can use in most of the models I'm trying to run anyway. And I like the fact that these are probably unlinked loci. So structure plot of my first sequencing lane sort of shows, this is like structure at k equals 8, I think. And it's basically showing that there's 2 clusters, and anything above 2 basically produces the same structure result. And those 2 clusters are most likely the 2 species, and I'll sort of map that later, and you'll see that. And then if you sort of pull out the two species and do some individual structure plots on those, at different levels, you find that there's what I'm calling sort of minor clusters. So the major clusters are probably associated with the species. Structure does not want to identify additional clusters, which is interesting in itself. You would often expect to find isolation by distance, and you're going to find 2 clusters if you look for 2 clusters. But structure just doesn't want to show more than 1 major cluster within a species. But it does want to show these sort of what I'm calling minor clusters. So these are groups of populations that seem to share something with each other, but it's a minor gene flow. And I'm curious to hear what people think about that, because it's a little bit iffy. So let's try mapping these structure clusters, major and minor clusters, onto my sampling map. So first of all, just mapping the 2 major clusters shows that they really do match the species distributions well, including sort of the populations that are a little bit mixed come out where you expect mixed populations to be. That's not surprising. Then if you sort of on top of that add these what I'm calling minor clusters, you find that it seems to be some geographic clustering going on. Not really strong. It's like, so like this is a cluster here that has some very southern populations and some sort of central California populations. But in general, these minor clusters seem to be some sort of geographic structure. And so I'm curious what's maintaining that? So at one level of structure, we've got these 2 species. And at another level, there's geographic structure that's not necessarily about who's closest to each other. So I guess if you go back to this, you can see that sort of 2 very close populations may not be in the same minor cluster. So we can do some sort of migration modeling. This is using div migrate, which is sort of equivalent to migrate n, but it's easier to run on genomic data sets. And it's sort of recapitulating the major clusters for the most part. It's showing, so these ones here with the blue dots in the center are the blue oaks, and the ones up top with the purple dots in the center are the two Oregon oak clusters that come out. And so that's recapitulating that, and it's showing that there's mostly migration sort of integrated throughout the history of the genome, mostly within those clusters. But then if you look at recent migration using Bayes-Ass, it's showing that actually a lot of recent migration is happening between clusters, and specifically between the Oregon oak cluster and moving into the blue oak cluster, which I'm not. My interpretation of this is that this is probably a pattern of recent migration happening throughout the history, and then those recent, then not introgression being allowed to occur. So it's sort of as alleles introgress from one major cluster to another major cluster, they're halted in future generations. So it'd be really nice to put this into a phylogenetic context, obviously, because that seems to be sort of the underlying story here. And so it'd be nice to have ways of inferring introgression at the same time as you're inferring splits in the phylogeny. And there are some ways of doing that. This is maybe not the best way. So tree mix sort of looks at all possible introgression events between clusters, and then locally optimizes splits in the tree around that. And it's showing a slightly different story. So the underlying phylogeny in the black lines recapitulates probably that sort of major clusters. And then the minor clusters are not really the same as I was finding with the gene flow models. And it's showing that there is sort of recent migration, so it's sort of between a very southern blue oak population and a very northern Oregon oak population. There can be fairly recent gene flow, and then there's sort of deep gene flow as well. So it's sort of near the bases of those major clusters, there's gene flow as well. And so I think with this that if you sort of believe the stuff I set out to prove, that I have shown that there is sort of there's a history of migration throughout these two, sort of this part of this clade. So I think that's evidence of this being a multi-species concept, or a multi-species complex. But I still obviously have a lot more work to do. I'm not super happy with the tree mix results. I'd like to get a better phylogeny to work with. My problem is that my phylogenies just don't converge. The longer I run them, sort of the more they just don't converge. And so there's maybe two approaches to that. One is sort of brute force. I just need to get onto a cluster computer and just run it for as long as I can. The second maybe option is to subset my data in some way, remove some of my samples. That sounds like a cop-out to me, so I'd rather do something else. If I can get a good phylogeny, I think these statistics, which people talked about earlier in this session, are probably a better way of inferring recent and ancient introgression. Recently, I haven't had a chance to try it yet, but recently I've been interested in hopefully using a multi-species network, Coalescent, which is actually using a model that simultaneously infers branching events and reticulation events. I think that would be the best, but it doesn't seem to work very well with genome data from everything I've heard. Second piece of ongoing work is to keep sequencing more samples. So far I've only sequenced 62 individuals from this clade. Ultimately I want 500 individuals. And then the third part, which is the part that I'm most excited about, is trying to integrate with the ecology. This is what Lee Van Valen would have wanted me to do. So this here, what I found is isolation by environment, but not geographic distance. That's the title of my ESA talk, which is going to be right here in this building in August. And so I'd like to integrate this fact that I have not found isolation by distance with these apparent minor clusters, which are shown some kind of geographic structure. And hopefully the answer to that lies in the third chapter, which is about the timing of flowering in these oaks, and how that might be controlling gene flow. Thank you.